Hello, and a very warm welcome to our Cell and Gene Therapy Insights Live 30 webinar. Rethink CAR T cell therapy with Scalable RNA Lipid Nanoparticle Platform. I'm Charlotte Barker, an editor at BioInsights, and joining me today we have two experts who will be exploring the challenges of current ex vivo gene delivery and editing strategies and the potential of RNA lipid nanoparticle platforms in cell therapies. So our first presenter is Dr. Rika Gatesy, who's Product Development Scientist at Precision Nanosystems. Rika holds a PhD in Pharmaceutical Sciences from the University of British Columbia, with high-impact publications at the interface of microfluidics and drug delivery. Her team is currently developing potent and efficient lipid nanoparticle compositions for cell therapy, currently focusing on CAR T cancer therapy. Also joining us today is Dr. Angela Zhang, a Senior Product Manager at Precision Nanosystems. After a PhD in immunology at the University of Toronto, Angela went on to specialize in immunotherapy and epithelial stem cell biology. She is passionate about enabling broader adoption of lipid nanoparticles by researchers and drug developers, particularly those working in cell therapies. After a presentation from Rika and Angela, we'll have a Q&A discussion. As always, do feel free to ask a question at any time throughout the event using the Ask a Question box, and we'll try to get to it during the session. Before we get started, I'd like to draw your attention to the Resources tab, where you can find links to get further information on the topics discussed today. And with that, I'll hand over to Angela to kick off today's presentation. Over to you, Angela.
Great, thank you so much, Charlotte. Okay, let's get going. Yeah, thank you so much for the introduction. We're very excited to have you join us today to talk about how to rethink CAR T cell therapy with a scalable RNA lipid nanoparticle platform. Specifically, we're going to talk about the application in ex vivo gene editing and delivery. We're especially excited because we have some very exciting new gene editing data we would really like to share with you today. And here's our agenda today. At Precision Nanosystems, it is our mission to accelerate the creation of transformative medicine that significantly impacts human well-being. And this is a mission that is reflected in the products and solutions we offer. And this all stems from our strong belief that genomic medicines are the future. And as you can see here, with the ability to silence, express, or edit genes, we're really able to target any genes in any way, making this type of medicine incredibly versatile and powerful. With the approval of mRNA-based COVID-19 vaccines and siRNA-based Onpattro, there has been increasing clinical validation, and with that, proof of concept manufacturing of genomic medicine utilizing lipid nanoparticle technology as a delivery platform. Now, when we talk about genomic medicine, just to get on the same page, we're really talking about a composition of cationic ionized lipid, cholesterol, helper lipid, and stabilizer. And within it, there's a nucleic acid payload. And in this presentation, we'll specifically be talking about payload of conventional mRNA or CRISPR-Cas in an RNA system. In terms of utility of this type of genomic medicine drug development, what you're most familiar with, perhaps is more on the left side, is the in vivo application, such as in the case of the COVID-19 mRNA vaccine and siRNA based on Onpattro. But this technology can also be used ex vivo to engineer, for instance, immune cells, and then these engineered cells can then be administered back into the human body. In terms of how LNP works, RNA LNP are thought to leverage an existing endogenous cellular transport pathway, specifically a low density lipoprotein or LDL receptor mediated endocytosis pathway as shown here. Once endocytosed, the change in pH facilitates endosomal escape, where the lipids interact with the lipids endosome, leading to the release of the nucleic acid into the cytoplasm. This is thought to be a gentle way to introduce genetic materials into a cell. And indeed, you would see data from Rika that we do see very high cell viability after RNA LNP treatment that is maintained even after sequential genetic manipulation. I do want to quickly dive into the in vivo application of RNA-LNP, that is COVID-19 vaccines. And I think we often talk about how quickly this vaccine was developed, manufactured, and approved. And there's a really good article from Nature that really explores the various reasons behind how this happened. While there's a lot of good reasons behind the speed, I think it is important to acknowledge that the mRNA vaccine platform itself played a big role in this rapid response. And that it's demonstrated to be safe and efficient, have relatively low cost, and it can be scaled effectively and quickly from a small preclinical scale to a very, very large commercial scale. When we think about how we can utilize this powerful technology for ex vivo cell therapy applications, here we're depicting two primary examples. Of course, you can, really be innovative here and really use this tools to build your cell therapy approach. But here we're just presenting two pretty simple examples here. On the left here is a straightforward delivery of, for example, something like a construct using LNP, which is kind of reminiscent of an autologous cell therapy. On the right here, this is a more multiplex or a sequential gene knockout followed by a gene delivery. This is more reminiscent of perhaps an off-the-shelf or allogeneic cell therapy. And you see data from Rika really giving examples of how LNPs can be utilized in both these applications and combination of these applications. When we think about delivery technology, viral vector-mediated gene delivery method is a very well-established method clearly and has been used, or it is used in current clinically approved cell therapies. However, as the fields move forward, especially when it gets into these multiplex gene editing type of applications for cell therapies, some of these known challenges become more of an issue, such as high manufacturing costs, potential insertional mutagenesis, and the fact that this technology may not be amenable to co-deliver multiple genes. So there is a desire in the field for a safer, perhaps less costly method that is also suitable for these multiplex genetic manipulation. So there's a lot of interest in, as a result of this, to look into non-viral options. One of the non-viral option that is currently being explored in the field, both in preclinical research and in some clinical studies, is electroporation. It is a good technology, but the biggest challenge is that it tends to be very harsh on the cells, leading to very low cell viability. And this low cell viability becomes even more of an issue when you think about these sequential genetic manipulation, or in the event where you want to dose your cells repeatedly. Electroporation is also not straightforward to scale. So there remains a strong desire for a non-viral method that's perhaps more suitable for these complex sequential genetic manipulation. So we have taken these considerations in the field and applied our extensive knowledge of both the lipid nanoparticles and the cell biology to design the GenVoy-ILM T Cell Kit for mRNA for the acceleration of T cell therapy development. It is an off-the-shelf lipid nanoparticle reagent kit so that you can get started right away. You don't necessarily have to be an expert in lipid nanoparticles. It is scalable on the NanoAssemblr microfluidic platform. And this is important especially if you're considering going to the clinic. That scalability becomes really top of mind. You see very nice data from Rika in several proof of concept study to show you that this reagent kit is optimized for complex gene editing and delivery in primary human T cells. Specifically, you see data around high cell viability that is maintained after sequential genetic manipulation. You see data around the very high efficiency for both knockout and expression. We have also done pretty extensive testing of this LNP formulation with downstream cell manufacturing workflow to ensure success, again, especially if you're taking it on for the first time. Finally, I do want to point out that if you're interested and are able to take your cell therapy programs to the clinic, we do have a team here at Precision Nanosystem that can talk with you about these non-exclusive clinical license. We won't talk about this at this presentation, but I'm happy to connect you with the right team members. Now, before I pass the presentation to Rika, I just want to take a moment to put this reagent kit in the context of what Precision Nanosystem has to offer for the full drug development path. So our lipid nanoparticle reagent kits are optimized to work on the NanoAssemblr instruments, as shown here. The kit we're talking about specifically is optimized on the Spark instruments right here, which is our smallest instrument that generates discovery scale materials. So we're talking about around 10 million CAR T cells worth of materials here. We have done the testing and we're confident we can scale this kit up to the Ignite scale, as shown here, so the one to the right, with sufficient materials for at least hundreds of millions of CAR T cells. So if you're interested in more information for the scale up, please do get in touch with me. And of course, you can see our instruments are able to scale all the way. We've done some of this studies both internally and with customers for in vivo applications. Finally, we also offer biopharmaceutical services that cover each stage of the drug development path that's shown here. So in the event perhaps you don't want to do this yourself, you want us to do it for you, with you, we have options there too. So feel free to get in touch with me on that. And with that, I will pass you over to Rika, who's going to show you some really exciting data.
Great, thanks, Angela. Great, so I'm here to present to you the gene editing data with the T Cell Kit for mRNA. But before I get into it, I wanted to get you excited about the field and what gene editing has done for CAR T cell therapy. Thank you. Okay, so I have some examples here on how gene editing is changing the landscape of CAR T cell therapy. And we can get enhanced potency of these cells. We can extend them to additional cancers and hopefully within the decade, extend them to solid tumors. We can reduce some of the side effects and importantly, as Angela has alluded, get to a stage of off-the-shelf manufacturing where CAR T cell therapy is available for everyone at earlier time points. And for all of these, we're looking at gene editing with the field now moving to non-viral delivery, particularly to electroporation. On the right-hand side of this slide, I put some current examples of common targets currently in clinical trials. For enhancing CAR T cells, we can knock out some inhibitory molecules like PD-1 and LAG-3 to extend them to other tumors like T cell leukemia. So to have a CAR T cell kill other T cells, we can simply knock out some common surface receptors found in T cells like CD3 or CD7. And then what our data is modeled upon is allogeneic CAR T cells or off-the-shelf T cells, where we can make a couple edits, particularly the TRAC locus and CD52, to reduce alloreactivity and eliminate graft versus host disease. But again, keep in mind that all of these examples are just examples, and that really there are limitless possibilities on how we can change the landscape of CAR T cell therapy through gene editing. The data here that we'll present, we have 4 distinct experiments to really show how we can integrate lipid nanoparticles into every aspect of CAR T cell engineering. In the first set, we'll show gene delivery. We have designed the second generation anti-CD19 CAR construct in a form of an mRNA that we deliver using lipid nanoparticles. Then I'll show two gene editing data sets, a single knockout in this example against the T cell receptor. We're using Cas9 mRNA and guide RNA, which makes it incredibly flexible to change the target sequence. And as we show with the double gene editing set, experiment number 3, we can combine lipid nanoparticles to knock out both the T cell receptor and CD52 for truly allogeneic therapies. And then we'll take it one step further and combine gene delivery and gene editing by making sequential edits for truly next generation CAR T cells. And just really briefly on our workflow, if you are in in a CAR T cell therapy, or if you have some T cells in your lab, you are probably very familiar with this workflow. You can either freshly isolate the T cells, or thaw frozen T cells. These are primary cells. Then activate and expand them for a few days. And then truly the beauty of LNPs comes into play in the next step. All we have to do is pipette the lipid nanoparticles into the flask or the wells. And there you have a final product. We have the CAR T cells, either that can be checked for protein expression, which is what we do using flow cytometry, or taken further for therapeutic applications. And really stay tuned, because in the last data set, we will do a co-culture assay with our CAR T cells, either with and without gene editing, to look at the therapeutic potential of the resulting CAR T cells. And as for the lipid nanoparticles, as Angela mentioned, we use the NanoAssemblr Spark. And this also takes just a handful of pipetting. In this case, we can mix the Cas9 mRNA and guide RNA with the T Cell Kit, the T Cell lipid mix, in a microfluidic cartridge, put it in the instrument, press a button, wait for a few seconds, and then collect the lipid nanoparticles. The particles here we used without any further purifications, and they are ready to go to be added on to the T cells. Right, so for the first data set, we are delivering the CAR mRNA to express the CD19 CAR protein. And in this case, we delivered using the lipid nanoparticles in orange, or with electroporation in blue. The top of the panel is detection at 24 hours, and then the bottom is at 48 hours. We can see the transfection efficiency on the left is significantly higher with lipid nanoparticles compared to electroporation, where we get 85% CAR expression with the LNPs and 55% with electroporation at both of these time points. As for the flow plots, we can see a much more homogeneous expression profile, where all of the cells in the population are very similar in their CAR intensity levels. And then importantly, on the right, we see the cell viabilities. Perhaps not a surprise to many of us that electroporation, the method itself, creates a significant toll on cell viability. And we see 90% cell viabilities with lipid nanoparticles as opposed to electroporation. So as you can see, we do have a very powerful technology here, both for for the transfection efficiency of delivering this gene, as well as maintaining high cell viabilities, both of them being important for the final cell yield of our CAR T cell product. Right, and now the exciting data set. Here we're doing a single target knockout using CRISPR-Cas9. We're delivering Cas9 mRNA and TCR-specific guide RNA using the lipid nanoparticles. So we treated the primary T cells on day 4 and then detected about 7 days after. And we can see a very strong dose response with the lipid nanoparticles, with our chosen dose being 3.2 microgram RNA per million cells in the subsequent experiments. We achieved about 75–80% single target knockout. And as you can see in the middle graph, we see a distinct emergence of the negative population after lipid nanoparticle treatment. And as before, we maintain very high cell viabilities, matching the untreated population at every single dose. And now we took it one step further. Here we're doing a double knockout where we packaged either TCR-specific guide RNA or CD52-specific guide RNA in the lipid nanoparticles. In this experiment, we treated with the 2 population of LNPs at the same time, the primary T cells, and then 7 days later detected the percentage of double negative cells. You can see that we achieved about 65% double negative population. And in the middle flow plot, you can see the emergence of this distinct population compared to the untreated cells. I think if I recall correctly, we did get 80% TCR negative, 70% CD52 negative, and then the combination of 65% double negative cells. And these cells are truly allogeneic and can be utilized for off-the-shelf CAR T cell therapy. And of course, as before, we do maintain very high cell viabilities, over 90% to the untreated controls. Of course, we took it one step further and we combined all the experiments. So I know this is a very busy slide. I did try to add in some methods in case you're interested, but I'll try to walk you through all the panels. In panel A, we have the workflow. So on day 4, post-thaw and activation, we added the gene editing LNPs. We waited for these cells to expand to really get sufficient numbers, and then did a TCR negative selection to deplete the TCR positive population. We took these pure gene edited cells and added the CAR mRNA LNPs onto them. And then the exciting data, we did do a sub-B15 killing assay where we checked the functionality of these CAR T cells. So in the bottom panel, we have the knockout and purification, where the starting sample had about 65% gene editing, and then we depleted it to greater than 99% purity. So this is the purple bar here that we then took further, and we expressed CAR onto them. We can see very high, strong CAR expression, 90% CAR expression with the unedited cells and 70% with the edited cells. And we also maintain high cell viabilities after all of these genetic manipulations, 90% to the untreated. And finally, we did the co-culture killing assay. In this case, we checked 2 different effector to target ratios, 1:1 and 2:1, and measured the specific lysis of the leukemia cells. What we see is there's functionally no difference between the edited and unedited cells, and both of these populations efficiently clearing the leukemia cells at the 1:1 and 2:1 ratios, about 80% cell lysis. So we're really excited about this data set, and it truly serves as a model to how we can integrate lipid nanoparticles for both gene editing and gene expression, and how fit these cells are to kill the leukemia targets. Right, so in summary, this data set really showed the ability of LNPs to engineer CAR T cells. Through CRISPR-Cas9, we show highly efficient editing that is tunable and can be multiplexed with a change of the sequence. We maintain high cell viabilities at every step, and these gene-edited CAR T cells are therapeutically highly functional. So when looking at the bigger picture, where do we go from here? Well, we see lipid nanoparticles advancing science and making an impact in cell therapy. But through scalability, and as we've seen with COVID vaccines, it's really only a matter of time before lipid nanoparticles impact therapies for us all. Thank you. Yeah, so thank you very much for listening. Angela and I will be happy to answer any of your questions.
The link. And I think all that's left then is to thank Angela and Rika once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Great. Thank you so much.